So up next, we have Dr. John Morris. He's a human geneticist and genome engineer. He earned his bachelor's degree in biology at the University of Western Ontario, then went to complete a PhD in human genetics at McGill University in the lab of Brent Richards. His PhD focused on identifying the genetic determinants of osteoporosis through genome-wide association study of bone traits in large biobanks. He's currently a postdoctoral fellow at the New York Genome Center and New York University in the labs of Neville Sanjana and Tulai Lapalainen, where he has developed single-cell pol CRISPR screens to study non-coding GWAS. John's postdoctoral work on linking GWAS variant to genes and through the method called STING-seq to expand our ability to understand mechanism underlying GWAS loci. John's achievements have been recognized by multiple agencies, as he was awarded both a CIHR Benting Fellowship, an NHGRI K99 Fellowship, and was awarded 2021 winner of ESHG Charles J. Epstein Excellence Awards. The title of his presentation is Beyond GWAS, Massively Parallel Genome Engineering to Pinpoint Causal Variant and Target Genes. Take it away.
Yeah, thank you so much, Yan, for that introduction. And I'm super excited to be talking in this seminar series. I noticed a lot of my friends have given talks in the series, so I'm delighted to be among esteemed company. I will also preface this by saying I am currently sick, so if I go into like a sneezing fit or like I start coughing, just please look away for a second. All right. So, you know, the work that I do is largely motivated by this question in genome-wide association studies, which is that, you know, many of these human genetic variants that are associated with traits or diseases tend to map to non-coding regions of the genome. So, you know, in this Manhattan plot, this is for a highly polygenic trait. And then in about 90% of these regions, it's not readily obvious what the underlying gene might be. And then so without knowing what the gene might be, it's hard to determine, you know, what the function might be. And without function, you know, then we have some difficulties in learning, you know, just fundamental things about the traits and the diseases that we study. And then so, you know, the problem here as we see it is that, you know, we do these genome-wide association studies, and this gives us this level of association. But, you know, how do we really go to the next step, to go beyond and to identify causal variants and their target genes? And then so the approach we're using here is to perturb these variants using CRISPR inhibition. And you just heard all about CRISPR activation, so we're going the other way, to identify those target genes and pathways. So the approach here is titled STING-seq, which is for systematic targeting and inhibition of non-coding GWAS loci with single-cell sequencing. And the first step is quite crucial. This is in our target selection, right? So what we want to do here is we want to be pre-selecting variants from our genome-wide association studies. And the first thing we'll do here is we'll actually find map them to identify causal variants. So for example, in a given locus, you might have like dozens of variants and you don't know which ones are causal. You can apply statistical procedures to try to tease out the ones that might be causal. And then what we're doing is integrating functional genomics data from whatever cell model or system of interest to these find map variants in order to identify the ones that might be functional. What we'll then do is design CRISPR guide RNAs to either target this, this candidate cis-regulatory element or directly target the variant itself. And then so what we do next is we target these GWAS variants with CRISPR in our cell models. So I'll talk more specifically about the type of CRISPR we're using and the type of cell model we're using. We also perform our experiments to a high MOI. So there's multiple guides being randomly integrated into cells. And then what we do next is a multimodal single cell capture. So this means that for each individual cell, we're capturing its full transcriptome. We're doing a direct guide RNA capture, so we know exactly which guides each cell received. And then we also profile for different cell surface protein levels. So we're not only going to be detecting changes in the transcriptome, but also in the protein level. And then we also perform cell hashing to just improve single cell data quality. And lastly, we do a similar procedure here as the previous talk, where we perform these pairwise tests for causal effects. So you can imagine a scenario where if you're interested in the effect of guide X on gene A, you might group all those cells together and you can evaluate, you know, based on the presence or absence of this guide, that the introduction of it was causal for the decrease in gene A's expression or in its protein level. So one of the takeaways here is that, when I'm showing you some of these results at the single cell level, I will be showing you gene expression, and I will be also showing you protein levels. Okay, so we wanted to build and develop STING-seq with these biobank-scale multi-ancestry blood trait GWASs. And so to do this, we use two different sets of blood trait GWASs. So what I mean by those are those of like red blood cell, platelet, and white blood cell counts. So for example, in a large population, you could take everyone's hematocrit levels. And then, this becomes a quantitative trait across a large population. And it turns out that, all of these blood traits are highly polygenic. So there are, thousands of GWAS loci to be studied here. The data we took them from are from the UK Biobank, specifically the Neil Labs GWAS Round 2 release, which is about 360,000 individuals. And then we also use data from the Blood Cell Consortium, which is about 750,000 individuals from 5 global populations. So in the end, we ended up targeting 539 of these candidate cis-regulatory elements from about 250 non-coding loci with CRISPR-I. And then so the cell model we're using here is a similar cell model that was discussed in the earlier talk, which is K562s. But we're using K562s here because they can actually be used to model human erythrocyte progenitor cells. So K562s tend to resemble cells that live at the top of this hematopoietic stem cell, this hematopoietic differentiation lineage. The type of CRISPR we use was, again, it was CRISPR inhibition or CRISPR repression. And the idea here is that, you have this dead Cas9 and it's fused to something that can recruit these repressor domains. And then typically studies tended to use this like single effector crab domain fused to the dCas9. A few years ago, when we were getting this project started up, it had come to our attention that using this dual effector vector should outperform the single effector lentiviral vector. And so we basically, we cloned this in the lab and we compared the two head to head, and we did see that this dual effector vector worked better. And then so for the CRISPR-I that I'm showing you here, we are using this crab and this MECP2 domain. Okay, so let's skip right into the results after we do our quality control, single cell sequencing, and differential expression testing. So as a reminder, we targeted about 540 of these candidate cis-regulatory elements from 250 non-coding loci. So this is our QQ plot. And then so the first thing I would draw your attention to is that our non-targeting guide RNAs are not significant here, right? So this means that, they shouldn't be targeting anything and they're not. So because of that, we can be confident in looking at our significant hits that are then deviating significantly from the line. And then so here we see that we actually have 135 of these CREs from 97 loci with 115 significant genes. We can break these numbers down a bit. So 16 of these actually came from population-specific GWAS variants. About a quarter of them, of the tested CREs, do have a significant target gene. And then I think what's most important here is that we can assign a target gene to a locus about 40% of the time. And then so, the takeaway here is that our expression tests are both well calibrated given the null non-targeting, and we can assign genes for 40% of tested loci. If you are interested in doing these types of analyses yourself, we have a companion paper that we published prior to this study getting published, which is called Scepter, published in Genome Biology in 2021. Okay, so now I'm going to walk you through the framework then for identifying target genes for GWAS traits, all right? So we're going to start off at this locus for corpuscular volume, which is a multi-ancestry GWAS. And you can see here at the top that we have a few candidate variants that are all within this credible set, all right? So when we look to see which ones map to enhancer-like elements, we see it's this RS48 in blue, which is actually the lead variant for the locus, and this RS12, which is the second most significantly associated variant at the locus. These two are both the top two most plausibly causal variants too from fine mapping. And if I'm just, studying a GWAS locus and I want to try to figure out what the gene might be, I'm going to integrate some orthogonal data sets. And then so one of those data sets we can look at are eQTLs, which is going to be, genetic variants that are associated with gene expression. So we looked at blood trait eQTL data, and we see here actually for both variants, they get annotated as eQTLs for this gene, MAPK-APK2. So just from this layer alone, I have a strong suspicion that the target gene for this locus is MAPK-APK2. We can go another step further and generate a separate orthogonal data set. So we also generated a bunch of high-chain data for the project. So this means we anchored on these H3K27 acetylation peaks, and we looked at all the promoter contacts for these peaks. And here we actually saw that it's only the red variant, which is the second most associated variant. It's only the red one that is mapping to any of these types of enhancer promoter loops. And in this case, it is looping to the MAPK-PK2 promoter. So what happens when we actually get in here and we perturb both of these sites with CRISPR-I? So here we see it is actually just the red variant that when we perturb this regulatory element here, we see a decrease in MAPK-PK2 expression, and we don't see any effect upon perturbing the blue variant. So what this tells us here is that, as we're studying GWAS loci, what we want to be doing is merging these convergent lines of evidence, and this can let us both identify what we think is the causal variant, which is the one in red, and the target gene, which is MAPK-PK2 in this case. A separate example is here where we're looking at another multi-ancestry GWAS, looking at monocyte counts. And here we see there's this huge block of associated variants. The fine mapping seems to bring us into this region here. And then this variant that sits at the top, RS74, is the most significantly associated variant and the most plausibly causal from fine mapping, all right? And then so if I turn to EQTL data again in blood traits, this time it's more pleiotropic. I see it's possibly CRY-BG2. I see it might be CD52, this SH3 gene, and the ZNF593 gene, all right? So now it's like, I don't really know what's happening. If I look at just 2D proximity in this case, I see that the closest gene body is actually CRY-BG2. The closest transcription start site is going to be UBXN11, and then CD52 is the second closest gene by either of these metrics. So when we actually get in there and perturb this element, what we see is that CD52 is overwhelmingly, the strongest target of this CRE, where CD53 sees a big drop in expression, and there's no effect on CRY-BG2 and no effect on UBXN11. CD52 also encodes a cell surface protein that was in the panel I used for the study. And then so I actually detect a significant decrease in CD52 protein as well as gene expression. And this has important implications for when we think about, what GWAS variants might be doing at these non-coding loci. You can imagine a case where if someone has reduced response to a drug target, it could be due to not because of protein damaging variations, but maybe due to enhancer damaging variations that are reducing their level of the protein. And in this case, I say that because CD52 is in fact a drug target. But I also mentioned this aspect of pleiotropy, where there are multiple EQTL target genes that are coming up. And then we actually see that here too, where the furthest gene in the EQTL set was ZNF593, and there's actually a small but significant effect on its expression as well. And then so this actually just gives us this more kind of complicated picture now that we should appreciate when studying GWAS loci, which is that in this case, we saw the strongest effect on the second closest gene. But given EQTL data and now perturbation data, it seems that this CRE has this regulatory pleiotropy, and that for some GWAS loci, we need to be considering not just the possibility of one gene, but multiple genes. And I'll also mention that, we also directly targeted CD52 and saw no effect on ZNF's expression. So it's really from just targeting this enhancer that you see this effect on this gene. So we can use these methods to identify target genes in cis. And what we do in fact find is that, with the previous example tends to be more of the exception than the norm, and that the majority of CREs do in fact target the closest gene, and that the majority of CREs target a single gene in cis, which is good, right? So this is a nice simple explanation. But it's worth paying attention to the cases where that is, of course, not the case. And that's where we see these, perturbation-like approaches being useful as these high yield discovery tools to be used orthogonally to other approaches to study GWAS loci. So now I'm going to pivot a bit and talk a bit about effects that we observed in trans, all right? So this is a case where we targeted, about 540 of these candidates. We saw 135 had significant effects in cis, and we saw further, 10 of those also had these widespread effects in trans. And then so when we look closer, so these are the QQ plots. Once again, you can see across these thousands of tests that the non-targetings are still null. We can see here that most of the networks we're observing tend to be due to, if you know your transcription factors, these are all transcription factors, GFI1b, NFE2, IKZF1, HEX, and ROX1, and also microRNAs. So this tells us that when we're perturbing these CREs, or these microRNAs, we're not only going to be identifying the primary targets of the CRIs, which are these transcription factors and microRNAs, but also secondary targets, which comprise these entire networks. So as we're digging into the networks a bit, one of the things I wanted to make sure of is that they are enriched for things that should be there. So to do this, I pulled together ChIP-seq data from K562, or I used target scan to look at the predicted target of a microRNA. And then so what this might look like is for this ChIP-seq data set, we have this peak here and we have this gene. So I would assign this gene to this transcription factor, essentially. And then so when I do this for my networks, I see significant enrichment across the board where we have GFI1b and V2. These are all significantly enriched for the things that should be there. And we also see this on the microRNA side with microRNA 142. But unfortunately, the RUNX1 and the microRNA 144 networks that we're observing are quite small. So you can see, we lack a bit of power to do these enrichment tests when the networks are too small. And then so for microRNA 144, we actually have some exciting follow-up spin-off projects in the lab to study this further. Okay, so, but essentially, this means that the networks I'm observing are enriched for things that I think should be there. So let's ask a more interesting question. Are these networks enriched for something else? Are they perhaps enriched for GWAS genes? And what would that mean? So to perform this test, I went back to all the fine mapping data. And what I do is I assign the closest gene to the fine map variants. And then by doing this across all GWASs, I can then group them together into just those that were like red blood cell traits, white blood cells, and platelets. And in doing so, I get gene sets for each of these trait classes. And then when I perform this enrichment test across the networks, here again, I see this broad enrichment across blood cell traits. GFI1b, for example, significant for white blood cells, platelets, and red blood cell genes. So what does this mean? This means that, as we're studying these complex trait genes, they're not only going to be modified by genetic variants that they have in cis, which is how they're included in this enrichment analysis, but also by genetic variation at transregulators. So something that is, modifying hex expression is also going to be modifying this, whatever white blood cell gene that is in this enrichment. This tells us that if we're looking at these data, that these downstream targets in these regulatory networks are themselves enriched for these polygenic contributions to complex traits. So the GFI1b network was the largest one. There's about 1,000 genes that we identified. So we decided to study it further just to see what else we can learn, because we did this experiment by perturbing these K562s. So we took our K562s and we look at just how these genes in the GFI1b network are co-expressed, and we perform clustering on them. And then so we saw 3 major clusters emerge, where clusters A and B tended to be comprised of genes that increased upon expression of decreasing GFI1b. And then cluster C, these genes tended to decrease upon decreasing GFI1b. So they're more like co-expressed. And then when we go back and we split up all these enrichment analyses by these different clusters, we see some patterns start to emerge here too. Where now we see the GFI1b ChIP-seq enrichment, which would identify the predicted direct targets of GFI1b, are exclusively within these clusters A and B sets, and not at all within cluster C. And that makes some sense because GFI1b is canonically a repressor. So what we've done essentially is by reducing or removing the effects of a repressor, these genes increased in response to that, whereas these genes in cluster C tended to decrease in response to removing a repressor. And then so because they're not enriched for the direct targets, this suggests that these are these further secondary targets of GFI1b. And then so we also see this pattern emerge with the GWAS gene set enrichments, where clusters A and B tended to be like the white blood cell and platelet genes. But then it's cluster C, or again, which is one of the weird ones, right? Because there's no direct targets of GFI1b, yet this is the one driving red blood cell enrichment. So we needed to now move out of our K562s, and we needed to move into a more of a primary human cell model in order to try to just understand, what is happening with these network genes and what else can we learn? So we shifted to studying human cell atlas bone marrow data. And we did that because of what we know about how GFI1b's known roles in hematopoiesis, where GFI1b is actually expressed in HSCs, hematopoietic stem cells, to maintain this like multipotent state. But then it's also expressed as it differentiates down into these red blood cell and platelet lineages. And then so when we look at human bone marrow data, which is this rich data set of these progenitor cell types, but also differentiated cell types, we see GFI1b is expressed in exactly the cells we would expect. So these like hematopoietic stem cells, these megakaryocyte progenitors, these red blood cell progenitors. And then what we do now is we take those clusters from our network, and what we can do is we can paint those genes onto this human bone marrow data, all right? And then so we see more patterns start to emerge that start to explain some of the things that we saw earlier with our enrichment analyses. So for cluster A, what we see here is that cluster A, their expression is limited almost entirely to these white blood cells. And then I'll remind you that these were the primary targets of GFI1b. So these seem to be genes that are important for white blood cell differentiation that GFI1b is actively repressing, all right? Cluster B, in like a reverse of cluster A, is only expressed in progenitor cell types and a broad set of progenitor cell types. These are also predicted to be the primary targets of GFI1b. And this also makes sense in the context of what we know, because GFI1b is also active in just maintaining this pluripotent, multipotent stem cell state. And then it's cluster C, which I think is one of the weirder ones, right? Because this is actually only expressed in red blood cell progenitors. And it is also the only one that was enriched for red blood cell GWAS genes, all right? But because there's no enrichment of the primary targets of GFI1b, we think these are actually the secondary targets of GFI1b that are co-expressed alongside GFI1b as it goes down these red blood cell differentiation lineages. So to just like zoom in a little bit further, just to give you a concrete example of this, we can look at cluster A, which is like the white blood cell cluster. And we can see, for example, there's this gene CD33. This is a GFI1b ChIP-seq peak. So GFI1b probably regulates CD33. We can see upon perturbing the GFI1b CRE in our data that we significantly increase CD33 expression. And CD33 happens to encode a cell surface protein, which is then in my screen. And then I can also see here a significant increase in CD33 protein levels. This is done. So this is in K562s, right? And then when we look at where these genes are expressed in human bone marrow cells, they're expressed exactly where you'd expect them to be, right? Where GFI1b is in only these red blood cell progenitors, and then CD33 is only expressed in these white blood cell differentiated cell types. Okay, so this tells us that we can do our experiments in these cell models that are good cell models for what we're trying to study. But then we can still go later on and integrate these primary human cell data to try to inform these findings from our cell lines. So I'm going to close off by talking about additional experiments that we did for the study. So I'm going to talk a bit about BeeSTING-seq for causal variant insertion. So this stands for base editing STING-seq. And so what we did here was we decided to switch out the CRISPR-I for base editors. So specifically here, we're using a cytosine base editor. So these will induce basically C to T or G to A GWAS mutations if we want. And we decided to use a paneless Cas9 variant called SPR water spry. And that's because, when we do these Cas9 experiments, we are limited by a PAM site, which is generally more fine for CRISPR-I and CRISPR-A because you're inducing some sort of like a window of effect. But for base editing, we do care about the explicit, precise location of the guide RNA. And then so some researchers found that, if you mutate the Cas9 enough, you can reduce this PAM requirement. And then essentially, this allows us to target almost anything in the genome. So we verified that the base editor works by targeting these CD46 splice junctions. And importantly, we use these non-canonical, non-NGG PAM motifs. So you can see here that we basically disrupted CD46 by engineering a single base allele change. And the PAM here was NAN and PAM here was NGN. So if this was regular Cas9, like this would not have worked. And then so this allowed us to screen 46 of the variants from our STING-seq target genes. And so let's dive into some results. So I'll give you one example. So this is how we can use BeeSTING-seq to identify causal variants, all right? So this is a locus where RS14 is nicely the lead variant, the most positively causal variant, and it maps to this, intronic enhancer element, right? So it is a C to T mutation in GWAS literature. And then so this is exactly the type of mutation that we can engineer with base editing. So we designed 2 independent guide RNAs here to engineer the C to T mutation. Now we can go in and we can compare this with our STING-seq results, right? So when we use CRISPR-I, we see, we see APP-BP2 as the target gene. We see that when we target that enhancer with CRISPR-I, its expression decreases. But now when we go in and actually engineer the GWAS effect allele, we see actually that there's an increase in APP-BP2 expression across these 2 independent guide RNAs. So this tells us that while we can still importantly use CRISPR-I in targeting these enhancers to identify target genes, which at the end of the day might be one of the only things we care about, we can go further and we can use base editing to identify explicitly the causal variants and in fact their affection of direct on gene expression, which is especially important in the absence of like well-powered eQTL data that would tell you something similar. So with that, I will wrap up. So I've shown you how we can use STING-seq for the study of GWAS to genes to function. So we perform these tests for causal effects in single cells. We can identify genes in cis and trans. And then I think one of the coolest findings here is that these trans regulatory networks are also enriched for GWAS genes. So this work was recently published in Science, so you can check it out. There's a bunch of cool other analyses that I had to leave out for time. One of them of which is like a power analysis. So you can figure out how many cells you would need to do this yourself for like expected effect sizes and based on gene expression. And with that, I would like to thank my postdoctoral advisors, Neville Sanjana and Tuuli Lapalainen. I'd like to thank my collaborators, various sources of funding, and I'd like to thank you for listening. So thank you very much. I'd be happy to take your questions.
There's some applause. Thank you very much, John, for this fantastic presentation. We have a question from the audience. I think this is related to power. Did you have power to test transcriptome-wide CRE, or did you have a look at cis genes, then test particular CRE for trans effects?
Yeah, that's a great question. So, I mean, when I did this analysis, because I have a GWAS background, so I'm very much one of those like, I don't have any prior assumptions, right? So I actually started off by doing full transcriptome-wide analyses for every single CRE. And then it was later on when I looked at every single QQ plot and I was like, wow, there are some that have like these clear effects. And there are others where there is like a clear point at the top and that's just the cis effect. And then so essentially, it comes down to when I look at an individual transcriptome-wide effect for a CRE, like, yeah, we are well powered for it. But, when it comes down to, like looking at, like reporting all possible trans associations, then you are going to run into power issues. So I only look at the ones then that I guess that I did find a network for. Yeah.
Okay. We have time for more questions. Let's see. Okay, another question from Matt Coelho. Great talk. Non-coding region can be repetitive. Did you have any trouble finding specific guide RNA to introduce your desired base edits?
Yeah, that's a great question. So yeah, it is a matter of not only that, non-coding regions can be repetitive, but also that, when you're base editing, you have so few options in the first place to engineer a given variant, because a base editor has a window of like 3 to 5 variants based on, whatever kind of base editor you're using. So the general advice in the field is to, for a given variant, try all possible base, which is only going to be 3 or 5 guides. And then, if you're interested in other things like, so I do filter out like large homopolymer motifs, for example. So yeah, there are going to be a section of guides that are just like, I can't design. If there's like a T terminator motif, then that just can't be designed. So yeah, basically, it comes down to a mix of both, right? So design all the guides you can, and there are some going to be still just like inaccessible due to these, repeat elements. Yeah.
So one more question here. Thanks for sharing your awesome work. Have you tried cell mixture STING-seq to see if cell type specific regulatory perturbation effect could be observed?
No, so that is some of the future work that we have planned though. So that's in the works, but yeah, that'd be a cool next step.
Okay, one last question, and maybe also in line with future works. Have you considered linking your GWAS to other kinds of traits? So, for example, neuro-related traits, or is it just difficult to get this type of cell line for your approach?
Yeah, no, so I don't think it's too difficult, because I am currently expanding into other cell lines, right? So there's a few angles you can take here though, right? Like from a GWAS perspective, you can look at whether, your, like in this case, blood trait variants might have some causal effects on neurological traits, right? Like you could look at like immune cell traits for effects on, and you can do this statistically through like Mendelian randomization types analyses, or you can just do like broad strokes with like LD score regression. And then it comes down to like, do you think you have the right cell types, right? So if you do think, you can study something about like a neurological disorder through the lens of, some sort of blood trait disorder, then that is something that you can address in these cell types. But then otherwise, yeah, it becomes a matter of, finding different cell types and then making sure that they're actually relevant for what you're studying. So like what Flo is showing, where they're using these like iPSC derived neurons, using her cell models might be a better fit, depending on, whether or not the GWAS variant actually maps to like these enhancers, and whether or not they have, you think they have these effects across different GWAS traits.
Perfect. So thank you very much, everybody, for attending. Thank you a lot for the fantastic presentation, John and Florence, sorry. Don't forget to tune in next month. Actually, next month, we skip next month, July, but next seminar will be August 1st. And we will have William Bakash from the Laboratory of Viral Diseases at NIH, NIAID, and Haider Inam from the Pritchard Lab at Penn State University. If you miss any part of the talk, it's going to be recorded and uploaded on YouTube for you to see. So thanks again, everybody, and see you in August.